Hello everyone and welcome to today's webinar, Applications of SV-AUC during the Development of Lipid Nanoparticles and Viral Vectors. I am Cassie Saltman of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. We encourage you to participate today by submitting any questions you may have into the Ask a Question box. We will answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Dr. Klaus Richter, Group Leader AUC at Coriolis Pharma Research. Dr. Richter, you may now begin your presentation.
Good evening. I'm happy to talk today about the topic of application of SV-AUC during the development of lipid nanoparticles and viral vectors. And I'm going to give this webinar presentation for the company I'm representing, which is Coriolis Pharma. Myself, I'm a head of the AUC team here at Coriolis Pharma. I've been with Coriolis Pharma for 6 years. And the company itself is coming from an analytical background and a formulation and lyophilization background with 17 years of experience, about 930 projects being done, 2,700 of those projects being performed. And we have 200 analytical methods in-house. We have a dedicated set of scientists and dedicated set of scientific supporters, and from those 200 methods I'm going to represent AUC today, and I want to show you a bit about analytical ultracentrifugation, how it was applied historically, a method with a very long history in biologics and supporting information for comparability studies and many other studies before, and how then the method became very useful now for analysis of characterization of viruses for gene therapy, and also for characterization of mRNA LNPs, which is the topic of my presentation today. At the very end, I want to speak a bit about how we can apply analytical ultracentrifugation during QC and product release, now that AUC is also available as a GMP method. A little bit of a background on analytical ultracentrifugation. It is a very different method from preparative ultracentrifugation. We use analytical ultracentrifuge to measure, characterize particles that are in solution and describe the sample that we analyze based on the experiment that we perform in the AUC. You see the rotor on the right side. The cells are inserted into the 7 empty slot positions. The 8th position is taken by the counterbalance. And as a zoom in, you see how one of those measurement cells is constructed. There are 2 channels in the measurement cell, one for the solution and one for the solvent. And they can be measured at the same time and then used to characterize the particles that are in the solution. The centrifuge is spinning, and while it is spinning, it is also measuring. You see the detection system on the left side, where there is an absorbance detection system. The xenon flashlight lights up at the top mirror, and then the light passes through the rotor, through the measurement cells, and is detected again at the bottom of the centrifugation chamber by a detection system. So while the centrifuge is spinning, the continuous measurement is performed to deliver concentration gradients of the sample in the solution. This is shown as a scheme here, where you see the sample on the right side and the blank on the left side. And while the centrifuge is spinning, there is more and more movement of particles to the bottom of the cell, represented here by time 1, time 2, time 3, and time 4. And at each of these time points, the detection system is recording a full scan of the concentration profile. And this is then represented on the right side here as the concentration profiles at the different time points. From these data, the whole set of all these data to up to 200 or more scans, the sedimentation velocity experiment is analyzed to deliver the mass, density, and shape dependency of the particles in the solution, and thereby deliver the concentration of different particles that are there and moving in the solution. The movement itself is analyzed based on the equation you see on the right side, where the most relevant parameters that we in the end extract are the sedimentation coefficient and the diffusion coefficient. And as a sum of all these different equations, the concentration of the individual particles in the solution. This is a complex and computationally intense characterization as this large data set needs to be analyzed. This method has been applied for a very long time and is used as orthogonal characterization in formulation studies, orthogonal information during analytical method development, comparability studies for biosimilar development, and stability testing to generative supportive data under GRP principles. As you will see during the talk, the method has unique features, can characterize unique critical quality attributes, which we feel makes it also important to have this method under GMP, and therefore stability testing of GMP batches and shelf life determination and batch and product release testing can be performed under GMP. Going into the method itself and how it was applied historically, analytical centrifugation has been a part of a large panel of different methods and was used for high molecular weight characterization and aggregation quantification. To give you a bit of a feeling of the data that are analyzed, this is just an antibody where we collect lots of scans, as you see on the left side, and each of these scans is 20 seconds apart, and you can see the movement of the antibody in the cell as the concentration profiles move further and further to the bottom of the cell, which is here. The data analysis then overlays this set of measurement data with a red simulation, and the red simulation is in this case matching almost perfectly the measured data, as can be seen by the residuals, to characterize the sample and determine the species that are in the solution. This is usually a very good precision that is measured, but still for those types of particles, AUC is competing with SEC-MALS and possibly field flow fractionation, which are also very precise methods. So 150 injections can be done here, superimposed, and that precision is certainly very hard to match, but unique features are coming from AUC experiments, which, as said before, make the method a highly relevant method for orthogonal characterization, support to these traditional GMP methods, and thereby contribute to the platform of methods that are used in comparability studies and beyond. Certain advantages for SV-AUC in these studies are that there is usually very good focus on the API in the solution. There's no matrix interactions, no membrane used. The particles are moving freely in solution. And as you will see in the further slides, this also makes the method particularly useful for the characterization of more complex pharmaceuticals. AUC methods have been qualified for a long time to characterize the method parameters according to the ICH guidelines, and the precision of high molecular weight species detection, the quantification of or the determination of a LOD and the LOQ, and the robustness against small changes to the setup make these methods well characterized and also applied during pharmaceutical development. Certainly, from the application of biosimilar development, there is a movement into larger and more complex APIs in the last years, with, in particular, viruses getting developed for gene therapy applications, mRNA LNPs getting developed as vaccines, And these APIs have particle characteristics much larger than the antibodies that have been characterized before. And as you will see, certain characteristics make AUC a particularly useful method to characterize these APIs. Just to introduce AAV, it's a very small virus, 20 nanometers in size, has a huge potential in gene therapy applications, and is used to bring in genes into patients that need this gene therapy for their recovery. You see the AAV on the right side. It has capsid proteins. It has a nucleic acid inside, and with the 20 nanometers, it is certainly one of the smallest viruses available. The need to produce these AAVs makes characterization of AAV samples highly important, but there are certain challenges to develop AAV products. AAV samples are composed of large assembled structures and costly in production. So usually the amounts available are limited, and therefore consideration has to be done during the AUC application to not use too much of these AAVs. A specific topic during AAV manufacturing is that empty and filled capsids are produced, and that the final product, of course, should only or mostly contain filled capsids. There is also partially filled capsids that are produced, as you will see, and AUC is particularly suited to characterize these partially filled intermediate species. The ratio between empty and filled species is a critical quality attribute, as it is important because immunogenicity is a topic, and one wants to minimize the amount of empty capsids in solution. SV-AUC is able to fast determine the empty-filled ratio, and there are even several AUC methods available that I don't want to get into to address this topic from different angles. In general, the best developed SV-AUC method at the moment is that for AAV characterization. So we did a short case study to characterize 5 different AAV serotypes with multiple methods to determine how well these different methods agree and how well these methods can be integrated into one study to characterize the empty-filled characteristics of particular samples. These different methods should, in the end, agree on delivering the critical quality attribute with highly similar results. So the approach we did is to characterize these with the different methods, and then to develop a standardized workflow for the methods to come to an agreement in terms of their critical quality attributes. The methods that we applied during the study are UV spectroscopy, negative stain electron microscopy, mass photometry, ion exchange chromatography, and SV-AUC. And just to show you how classical SV-AUC results would look like for the 5 different serotypes and the different samples that we are having here for the study, the empty species for the 5 different serotypes is highly similar. There is almost no difference between the different serotypes. The filled species likewise is highly similar, but some of the AAVs that we had during the study have more or less empty species still remaining. And the in-process controls that we investigated have both empty and filled present at the same time. So the goal was to determine the empty-filled ratio with the different methods and to see how good of an agreement we may be getting when comparing the outcome of these 5 different experiments. In general, analytical ultracentrifugation, as shown already, delivers the empty species, intermediate species, and the filled species. While for the other methods, the intermediate species is less visible. For the other methods, ion exchange chromatography can be used to see both of the peaks as an empty and a filled species. UV spectroscopy just gives a spectrum that moves between 260 and 280 nanometers based on the based on the percentage of filled species. Electron microscopy delivers particle images that contain either the nucleic acid or not, and therefore take up stain or do not take up stain. And then the analysis is done based on the counting of the particles, and mass photometry determines the mass of the particles based on the interferometric contrast, and thereby delivers a empty and a filled species on a scale of the mass of the particle. When looking at the output of these methods, it became clear that we cannot immediately take the result of each of these methods and compare directly. And the reason for that is very straightforward. The negative stain uses a counting approach that leads to a direct empty-filled ratio based on the number of particles counted. The same is true for the mass photometry, where likewise the counting approach delivers a direct empty-filled ratio. The other methods are based on absorbance, and here it now is it needs to be taken into account that the extinction coefficient of the empty species is much lower than the extinction coefficient of the filled species, especially when wavelengths like 260 and 280 nanometers are used, and therefore the correction has to be done to come to individual particle numbers based on the absorbance numbers that are determined directly from the method. UV spectroscopy has its own calculation that we developed during that study here, and it needs to be based on the 260, 280, and 230 nanometer contribution and the individual contribution of the empty and the filled species. So in general, when looking at the different wavelengths, at 230 there is about a 1.3-fold higher absorbance of a filled species versus an empty species that needs to be corrected for. At 260 nanometers, there is a 4 to 7-fold higher contribution of the filled species due to the absorbance of the nucleic acid, depending on the size of the nucleic acid that is in the capsid. And at 280 nanometers, there's a 2 to 4-fold higher absorbance for the filled species. So this needs to be accounted for, and the way to account for is based on response factors that originate from a comparison of the different extinction coefficients. And with these response factors, one can correct the original numbers obtained directly from integration of the absorbance signals, and correct it with these response factors and these equations to get the number-based empty percentages, partial percentages, and filled percentages for SV-AUC. The same approach has to be taken for ion exchange chromatography, where likewise the filled AAV capsids give a much higher signal than the empty capsids. And if we want to compare that to the electron microscopy or mass photometry output, then we need to correct for these different extinction coefficients of the particles. For UV spectroscopy, this equation is fairly complex, and one can calculate the concentration and the percentage based on the comparison of 230 to 60, to 60 to 80, or to 30 to 80 nanometers, and thereby come up with a number of capsids that are derived from the spectra. Looking at the agreement between the methods, when plotting AUC on the x-axis, one can see that, especially with the ion exchange chromatography, there is a very good agreement, and only in some cases, in this case a slightly aggregating sample, this agreement is diminished. But in general, the agreement between those two methods is very straightforward. Same is true for mass photometry. where there likewise, after the correction for the absorbance differences, this agreement is very good between mass photometry and SV-AUC. Agreement is a little bit less good for UV spectroscopy, where, for example, impurities in the preparation affect the UV spectrometric approach dramatically. In this case, it is residual presence of iodic xanol, where the spectra then obviously are affected by the presence of these impurities, and then these field species do not deliver the same data quality. Also, for electron microscopy, the scattering around the line is a little bit higher, and it may be better the more particles are counted in electron microscopy. And but in general, it is possible to bring all these methods to a very good agreement, especially with ion exchange chromatography and mass photometry. AUC agrees very well. So in general, this approach allows to use multiple methods in, for example, comparability studies where all of them report on empty and filled capsid ratios, and work with those methods so that the empty and filled capsid ratio is in agreement between the different methods. In a recent study, we tested whether this can also be extended to the filled species in the case of mass photometry, where we used a AAV sample that has only a short DNA strand as filling up the capsid, and if the DNA strand is short enough, then two of those DNA strands can fill the capsid, and therefore we get a profile with an empty, a single filled, and a double filled species. And when comparing the SV-AUC approach at 3 different wavelengths with the mass photometry approach, then we come to a very good agreement of empty, single filled, and double filled species. So apparently it is possible to extend that approach also to a system where there is 3 different AAV species present. To sum up on AAVs, SV-AUC can be applied as an orthogonal method for empty and filled ratios together with other methods. The correlation of the data is very good, and SV-AUC is unique in that it provides information on impurities and concentration of AAV species at the same time. Looking at the size range that we have been working in at the moment, we see that AAV is at around 20 nanometers and a very small virus type. But there is also other virus types that are developed as pharmaceutical products, be it adenoviruses or lentiviruses. And we aimed also to understand to what extent these viruses can be used for characterization of samples, and what role AUC may play in the characterization of these samples. You see the size range and the different methods that are available in these size ranges. And we aimed for a case study with lentiviruses, and lentiviruses are 80 to 100 nanometer size gene therapy application. They are often used for TCAR virus and in vitro modification of cells that later on then are used as therapeutic products. The formulations can be complex, and we will see that also in the study that I'm going to show, that there is, for example, serum albumin in the background, which makes it not easy to study those samples. The virus samples likewise are in limited amount. And for those larger viruses, it's very hard to work with chromatography solutions, only if specific columns are available that allow the separation of products in that size range. SV-AUC can be adapted straightforward by using a very low speed to characterize the samples. So in this study, our aim was to characterize 2 lentivirus samples with also multiple methods, and to apply these different methods to determine which one is best suited to characterize the lentivirus samples. The approach was to use 2 production samples of lentiviruses, one being adherent, lower concentrated sample, and the other being a suspension sample with a much higher concentration, but also with some aggregation issues to be characterized. Looking at the different methods, each of them struggled to some extent to characterize the material 1, which is the suspension culture, and the material 2, which is the adherent culture. And as you see, the material is fairly turbid, so samples were diluted substantially for the characterization of the particles. Looking at the results of SV-AUC, we decided to use a two-step approach. In the first step, SV-AUC was performed at a very low rotational speed, and there is already a difference visible between the two samples. The aggregated LV material shows also some aggregation here in the higher molecular weight range, even at the lowest speed, and only a very small amount of nicely characterizable virus, while the less dense material of the adherent culture shows a sedimentating species here, which is the virus, plus then also additional impurities. When looking at the profiles, there is rarely any monomeric lentivirus visible in the material 1, while there is a clear peak at around 650 S of lentivirus species visible for the material 2. To characterize the formulation completely, we accelerated after the first speed step into a second speed step at 42,000 RPM, and then sedimented all the remaining particles in the solution, in this case the serum albumin, and characterized then these two speed steps independently of each other, but with a second speed step taking the first one into account, as it is clear that the first sedimentation step already led to some sedimentation that is still visible in the second step. So one cannot completely ignore the first step, and SETFIT gives an option to do this. And this speed step analysis was applied, and in this way one can characterize the lentivirus in a first step, and then when it is a complex mixture, all the other relevant species that are only visible at the higher speed in a second step. This way, massively expanding the window where AUC can be applied, and the lentivirus can be characterized at the same time in the same run as the formulation components and BSA. With that, I want to come to the applications that we have for mRNA LNPs. And these mRNA LNPs likewise are structures in the nanometer, 40 nanometer, 100 nanometer size range. They are complex build structures made out of lipids and mRNA inside the lipid structures. And this complexity also makes it important that we use methods that are not disruptive to these structures. We can adapt the speed window to the LNPs, and in similarity to mRNA LNPs, we can also characterize liposomes, empty LNPs, LNPs filled with siRNA. In general, the characterization of these lipid-containing structures is challenging due to the heterogeneous composition, and also due to the potential interaction of lipids with matrices, be it chromatographic matrices, or be it membranes for field flow fractionation. We did a short case study where we applied stress conditions to LNPs and tested different stress conditions. The most drastic change we saw with freeze thawing, and in this case we used DLS to just get a rough overview over the changes that we had introduced to these LNP structures. You see that the freeze thawed LNPs got much larger in DLS, while the others, there is possibly changes, but it is not clear to what extent these changes are consistently visible. When looking at AUC, we applied an SV-AUC method to characterize LNPs. The untreated LNPs are visible on the left side. And the first interesting feature of LNPs is that they are lighter than the formulation buffer and therefore float. So while all the other particles so far sediment to the bottom of the AUC cell, these particles move to the top of the AUC cell, and therefore the scans that are visible here show concentration profiles that get lower and lower with time at the bottom of the cell, and the particles move to the left side, which is the inside of the rotor. After vortexing the LNPs, there is higher molecular weight species visible, and this can be analyzed by using, in this case, the software Ultrascan, which is able to simulate the movement of these particles and also take into account that there is larger particles present here. And the resulting simulation scans match almost exactly the profile of the measured data. Looking at the freeze-thaw process, this sample is very different than the samples before treatment. And still, even under those conditions, it's possible to fit these data with Ultrascan and obtain a species distribution that reflects the changes of these changes that were made to this sample. Changes are visible here in the distribution, where the freeze-thaw sample is a very broad distribution of different species up to very large sizes, while the other two samples are defined, give a defined peak, all of them at a negative S value, and all of them floating to the top of the solution column. Quantification of these species can be done based on the distribution, and this quantification can be performed in replicates to obtain error bars. And then the freeze-thaw clearly has a high molecular weight species, way higher than the other samples, but also for the vortex, it becomes visible that there are changes. For the high temperature stress, there are changes. And when looking at the sedimentation coefficient of the main species, also their changes are visible. And the main species, which is barely present for the freeze-thaw stress, seems to be becoming also a bit larger. So LNPs can be characterized based on the densities that they have and based on the size. And both of these parameters contribute to the flotation speed of the particles. They would be moving faster if they are lighter in density and if they are larger in size at the same density. If we do changes to formulation or to solvent compositions, this could have a substantial influence on the sedimentation properties. And this also leads to a method which allows us to characterize LNPs in more detail, focusing on the density of the particles and focusing on the question whether all the particles in the solution have the same density. And this can be done by modifying the formulation buffer by using D2O instead of H2O, different ratios, and thereby modifying the density of the formulation buffer. This has been published in 2021 by Amy Hendrickson from Boris Demeler's lab. And in this case, 4 different D2O concentrations were used, and the influence of the D2O concentration on the sedimentation coefficient is visible in these boundary fraction plots, where the light water or the normal formulation buffer is in between a slightly negative sedimentation coefficient and about 100S. Then 40% leads to faster flotation. The buffer is getting more dense, and therefore the particles float faster. The negative sedimentation coefficient gets bigger in value, and at 70% even more, and at 95% D2O even more. When all of these are when it is when the calculation is performed in a way to match all these densities, then the density distribution can be obtained. And this was performed in that study for different kinds of LNPs, siRNA and BSA and individual RNAs as shown here. So for the LNPs, it is clear that these are around a density of 1. And the nucleic acid is very heavy here at a density usually in the range of 1.6. We also apply this method routinely to characterize LNP types and produce density distributions like this, where in this case 2 different LNP types were compared, one of them having a fairly consistent density throughout the sample and only a little bit of lighter particles in a lower percentage range. The other LNP type that was characterized here is a bit lighter in density and then also has a substantial part of particles in the range of 20% as visible here on the boundary that are lighter than the particles, the remainder of the particles. This is done in a stepwise approach, the analysis. One can first look at the sedimentation coefficient distributions at the different D2O levels, use those to come up with an average density of the particle that would describe the whole average particles in the solution, and then move towards the density matching part of the analysis and produce the density distributions that have the percentages of empty or possibly differently filled LNPs in the y-axis. Summing up over the product-specific requirements that we are having for the products, proteins, peptides, and mRNA and monoclonal antibodies, the size in the range of 1 to 300 kilodalton can be covered depending on the speed that is applied. Contents of monomer, dimer, LMVS, and high molecular weight species are derived from the analysis. Orthogonal methods to AUC are sec mols, or AF4 mols. And the QC perspective here is that sec covers a lot of those questions, and it will be interesting to see what role AUC plays now that it's available also under GMP. Adeno-associated adenoviruses and lentiviruses are clearly products in a perfect size range for slow speed or low speed AUC experiments. It is a question assembly status loading with nucleic acids. There are orthogonal methods available also, sec-mols, AF4 mols, but AUC provides good resolution and good precision for those particles and also can deal with the size of these particles. For LNPs, likewise, these nanometer-sized particles are in a very good range for analytical ultracentrifugation, and the very nature of these LNPs makes them perfectly suited to perform AUC. When looking at the QC perspective of AUC, it is so far clear that during early development and during GRP studies, the focus is on a solid, well-understood method, scientifically sound method, and a method that can be qualified to a certain extent, and with the application of good documentation practices, written reports and protocols, orthogonal methods can be supported with AUC, and comparability studies can be supported with AUC to characterize the similarity, for example, of particles and of APIs. Under GMP, now we need to make sure that the method is very well understood, validated method, GMP being required for phase 1, 2, and 3 studies to characterize APIs for human use require now also a full validation and a protocol, a full validation report, and a fully validated method. And to come to that state, it is important that the whole workflow of AUC experiments is validated and qualified. And this, of course, involves a lot during data analysis, so that the software and the audit trail and many aspects of the data analysis fulfill the requirements needed for a GMP study, but also the part of the workflow that precedes this, the experiment, the sample preparation, the sample receipt, and the preparation of the instrument are needed to be monitored, documented, and fulfill the GMP criteria. And this whole workflow has been implemented now at Coriolis, working with data analysis, both ZFIT and ultrascan. So we believe that the GMP principles being followed at every step contribute a lot to AUC becoming a GMP method applied for the different kinds of APIs, likely in the viral range, likely for LNPs. And this workflow of multiple steps needs to be controlled and ensured to be compliant with cGMP. There's a challenge to develop the tests that make every step that is used during this process controllable. And what we aimed for is a flexible workflow to either work with ZFIT or ultrascan, and also to accommodate larger viruses and workflow under BSL-2 conditions, as lentiviruses require, for example, BSL-2 conditions. With that, I want to acknowledge the people that contributed to this data at Coriolis, our internal studies that were performed with the help of the scientific team around Andrea Have and Tim Manson, and many other people in our scientific team and within my team at Coriolis Pharma. Our scientific advisors, Gerhard Winter and Gideon Kersten, our cooperation partners for the lentivirus study, Klaus Wuchner, and for the AAV studies, our cooperation partners at the LMU and at Beckman, and our AUC group at Coriolis Pharma, and all our collaboration partners. With that, I'm done with my presentation, and I'm happy to take your questions, and now proceed to the Q&A session.
Wonderful. Thank you, Dr. Richter, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box located in the lower left corner and click Submit. We'll answer as many of your questions as we have time for today. Our first question asks, seeing the comparison of AUC to SEC and AF4, do you think AUC could be used in ADC or antibody drug conjugate work? If so, what could AUC bring in regards to AUC?
Yes, thanks for that question. I think indeed that AUC can contribute here quite substantially. First of all, AUC already contributes to studies where proteins are investigated, and that, of course, doesn't change if that product is an ADC. But also there's an additional question for ADCs, which is the coupling ratio of the drug conjugate to the protein. And I think, depending on the construction of that conjugate and the possibility that this drug conjugate has specific absorption properties, it could be that methods are turning out very valuable where one looks at the conjugated drug with one wavelength and the protein moiety with the other wavelength, and thereby compares. It needs to be seen, but it's an interesting system that is a bit more complex than the protein, and usually AUC is very good for complex systems.
Excellent, thank you. All right, our next question here says, is the speed step AUC centrifugation all done in one experiment, or does the centrifuge need to be stopped between the speed steps? And is there any sample preparation that needs to be done?
So it's all done in one experiment. The centrifuge can be programmed to run first speed steps at 3000 RPM and then accelerate to a higher speed step. So there's no stopping in between. The stopping would also disturb the concentration profiles. So the sample would not be simulatable to that extent anymore. And therefore, it's important that we don't stop in between. We run the first speed step, sediment the larger particles, and then accelerate to sediment the remainder of the sample. The software can deal with this. It knows the two speed steps. It simulates the first part and then starts with the analysis of the second part. So we don't stop. Regarding the sample preparation, AUC is done under optimal conditions in the formulation buffer. If that is possible, then the formulation buffer is the best diluent that we have. And that also is a bit of a unique feature of the AUC in this situation, because we don't need a mobile phase, and we dilute, we put the sample into the measurement cells and we spin, and under the best conditions, there is no other step involved in that process.
Okay, excellent. Thank you. Our next question that we have here states, LNPs are quite heterogeneous and many other techniques seem to have trouble identifying the entire heterogeneity range. Sorry. Can AUC help me better understand my polydispersity?
I think it can. I think what we showed is that there is different approaches to AUC. One can just run one sample in formulation buffer, basically as shown initially, and compare different kind of stressed versions, different batches, different LNP types directly against each other. Or one can go a step further and also look specifically at the density and the heterogeneity within one of those samples in respect to that density of the LNPs with a D2O approach. And I think so far that is a fairly unique approach that AUC has here. And I think this could turn out very valuable when trying to understand the LNP composition or the process of making those LNPs and the consistency of those processes, for example.
Wonderful. All right. We have a few more questions here. Again, if you have questions for our speaker today, go ahead and submit them into the Ask a Question box, and we will try to get to as many as we have time for. Our next question here says, the LNPs and the lentivirus samples cover a large sedimentation coefficient range. What does this mean, and does it affect the ability to analyze the samples or understand their characterization?
Yes, so that was indeed visible in our lentivirus sample. Certainly, if the whole of the sample can be covered within one speed window, then it is good to run only one speed. But if there is other parts of the sample or interesting parts of the sample that need to be further investigated at a higher speed, then the speed steps model is needed to address this whole range of sedimentation coefficients that are represented by the particles in the sample. Lentivirus at around 650S is a fairly large particle, especially if then there is also aggregation of further lentivirus, even at larger S values. And this window needs to be made large. And therefore, it's good to have a powerful software solution that can deal with this large window of sedimentation coefficients at a sufficient resolution. And that's, I think, the key to analyzing those samples then.
Wonderful, thank you. All right, and our next question here says, in your quantification of LNP and large virus aggregate species, how do you correct for MIE scattering, which is highly skewing for the large particles proportional to the sixth power of the LNP radius, giving you a distorted quantification.
That's certainly a topic when dealing with larger particles. So the AAVs do not have that problem to this extent. They are still a clear solution with a very limited amount of scattering. But the larger particles then do scatter quite substantially. Actually, in the studies that we performed here, we did not correct for that scattering. We used it as part of our quantification strategy for the integration, and it is a descriptive parameter of the sample. We could indeed correct for it. The question is whether that would be supportive for the robustness and the repeatability and precision of our method. Therefore, to compare a batch, we accept here that there is a scattering component, but as it is the same for all the batches that we analyze, we take it into our quantification. If that, I hope that is sufficient for that explanation. Let me know if there's more to it.
Wonderful. All right. We have time for a couple more questions here. Our next question asks, what is the drawback in analyzing finished drug product compared to API?
I don't see much of a drawback for the liquid product. We can work with a finished drug product. We often work with drug product samples. In many cases, they are different from the drug substances. They have a different formulation buffer. They have a different concentration. In many cases, we compare directly drug product to drug product samples. So we do not have to worry too much about the differences of those samples to each other, the drug substance and drug product. But in principle, both of them can be analyzed similarly. So I don't see a drawback working with drug product versus API.
Okay, excellent. Thank you. We have time for one more question here before we wrap up. This last question asks, what are the sample preparation steps required for AUC, and do you find these to be extensive compared to other methods?
So in traditional absorbance analytics, when using the absorbance detector, our main preparation step is in many cases a dilution of the sample to bring it into the range that the absorbance detector can deal with. This would be roughly an OD of about 1. And for example, for a high concentration antibody formulation, that's a substantial dilution step that we need to apply here. High concentration formulations can come with 100 or 200 mg per ml. And we need to go down to 0.5 mg per ml to use the absorbance optic at 280. So this dilution step is mostly what has to be done to use the absorbance optic. There's a workaround using the interference optic that can be used at much higher concentrations. I don't think we can reach 200 mg per ml as for high concentration formulations, but we can go substantially beyond the 0.5. But then also dilution step in the right dilution buffer or in the correct dilution buffer would be the only requirement to perform the AUC experiment. Beyond that, we need to prepare our measurement cells, make sure that they are clean, align them in the rotor, and then we can put the rotor into the centrifuge and start the spinning process. Compared to other methods that also investigate that kind of APIs, that would require a mobile phase to be made, and then has an auto-sampler that is also taking the diluted samples. So I think we are talking about a similar level of preparation and throughput that possibly is we can come up to 14 AUC samples in a single run, and a run usually takes between 8 and 24 hours.
Wonderful. I want to thank you again for your time today and your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.